Okay, you might remember that before break, we were talking about DNA, short for deoxyribonucleic acid. And James Watson and Francis Crick, who rhymes with prick, did I say that? With major assistance from Rosalind Franklin, worked out that this molecule that's present in all living cells, in the nucleus of all eukaryotic cells, is a very long rope-like molecule. If we could take all of the DNA out of one of your cells and put all of the pieces end to end, it'd be about 6 feet long. And it's shaped like a double helix, two strands twisted one around the other. As we've said, DNA is a polymer, meaning it's made up of a long chain of similar molecules that link one after the other. The subunits that make up the DNA polymer are called nucleotides. And each nucleotide has a sugar and a phosphate. The sugar is just a ring of carbons, hydrogens, oxygens. The phosphate is a little cluster of a phosphorus and some oxygen atoms. And then you have one of 4 bases. The analogy I always think of is if you think of a polymer as being like a train, all of the cars on the train have the same wheelbase, right? The same wheels at the same distance from each other. They've all got the same couplings that hook them together. But what goes up on the top can vary depending on whether you've got a box car, a flatbed car, a tanker car, a refrigerated car, whatever it might be. So just as all train cars on a train have the same wheels and the same connections, but different stuff on top, all nucleotides have the same sugar and the same phosphate, but they carry different bases: adenine, thymine, guanine, or cytosine. And DNA is a polymer of these, or to be more precise, it's 2 polymers wrapped around each other. We talked about how DNA can replicate because those 2 polymers can unzip from each other and each be used as the pattern to make a new strand. But where the rubber really hits the road is that there's something critically important about the order that the nucleotides are in. There were people that hypothesized back in the early 50s that every piece of DNA would just be ATGC, ATGC over and over again. And in fact, it's not so. The order of nucleotides it doesn't look like there's any obvious pattern to it. You can have long strands. I see there's a GGG, GGG in there, and then there's others that vary a lot more. And what's important about this is the order that the nucleotides are in carries information. Just like the order that letters are in a word carries information. And we now know the order of 3.2 billion nucleotides that make up the total set of human genetic material, or as we call it, the human genome. To read that order, what we have to do is study a second major class of molecules called proteins. And every living cell has to make thousands of different proteins to carry out whatever it's doing, its metabolism, whatever role it might be in the body that it's a part of, whatever the case might be. Sorry, checking the chat here to make sure you put your name in the chat if you have not done so already. There we go. And there are proteins that are important in structure, like the collagen proteins that make up your tendons and ligaments, like the Achilles tendon you can feel at the back of your heel. That is tough and stretchy and springy because it's made of a particular protein called collagen. There are proteins that are important in enabling cells to move. Proteins called actin and myosin are what make your muscles able to contract. Enzymes are proteins that make chemistry happen, that actually drive your metabolism, direct it. You have signal proteins like insulin and transport proteins like hemoglobin in your blood, and lots more. There's a ridiculously large number of proteins that cells in your body make. Different cells making different proteins, I might add. Proteins are polymers of a different type. They're polymers of a monomer called amino acids. And I promise you, if it weren't for proteins, it would be a amino world out there. Right. There's actually many thousands of amino acids, but there's only 20 that are used to build proteins. Here's where we're getting at this. The order that you put the amino acids in determines the shape of the protein. A protein shape is critical for determining what that protein does. If your muscle cells did not make proteins called actin and myosin with exactly the right shape, they would not be able to bind together in such a way that under the right conditions, they can make your muscle contract. All right, those proteins that make your muscles twitch are able to do what they do because they're in a particular shape. The order that the amino acids are in is what determines the shape of a protein. And the information that's carried in DNA is the information on how to arrange amino acids in the right order. If you inherit a piece of DNA that carries faulty instructions for assembling one of your muscle proteins, that muscle protein will have the wrong shape. It won't function, or at least it won't function as well. And if you survive, you'll probably have one of several conditions called muscular dystrophy. So as I mentioned, there are thousands of amino acids. Red Bull, if you look on the can, it says it's with taurine. Taurine is an amino acid in Red Bull. It's not one of the ones that you use to make proteins. Anybody who is into athletics might know about supplements with compounds called ornithine and citrulline. You can buy these at like the general nutrition centers, you know, these stores that sell nutritional supplements and things like that. I don't really know how effective they are. Some of you would know better than I. But those are examples of amino acids that are not used to assemble proteins. They can still be important. They still may play a role in your metabolism, but they're not one of this 20 group of 20 that are the building blocks of proteins. They've got other roles to play in metabolism. So what makes an amino acid is, just like every nucleotide contains a phosphate, a sugar, and a base, every amino acid has three major parts. The amino group is an atom of nitrogen stuck to a couple of hydrogens. The carboxyl group is a carbon with a couple of oxygens and a hydrogen for seasoning. The aminos in blue here, the carboxyls in yellow. And what makes amino acids different from each other is they've got different R groups. I think I've already made this joke, but amino acids are especially important to pirates because they have R groups. Thank you. And what the R group is determines which of the 20 amino acids you've got. The simplest R group is shown over there at the left. That's a simple hydrogen atom. I believe that amino acid is the one called glycine. The one on the right has a much more complex R group that is a chain of carbons, hydrogens, and nitrogens. I think that one is valine. I could be wrong about that, but those are just examples of different R groups. And it's the different R group that makes amino acids different from each other. So by all the gods, do not try to memorize any of this stuff. This is just chemical diagrams of the 20 main amino acids. For each one, up at the top, you can see that there is a carbon. On the left, there is a 3 hydrogens and a nitrogen. In this case, that's the amino group. There's a carboxyl group to the right. There's a little extra hydrogen at the top that's just kind of there. And then the shaded areas are the different R groups. And you can see that some of the R groups are very small. Some of them are phenylalanine. That's actually a ring of carbons and hydrogens. Tryptophan has a double ring. Here's some more. The shaded stuff, again, is different R groups. Glycines at the upper right is very tiny, and tyrosine at the lower right is a large R group. And here's the rest. In all of these, the R's are shaded. And again, you certainly don't need to memorize the names, and you definitely don't need to memorize the formulas unless you want to. Now, what happens is that amino acids will form a bond between the amino group of one amino acid and the carboxyl group of another. So on the left, in red, we have an amino acid that happens to be called aspartic acid. On the right, in blue, we have an amino acid called phenylalanine, and we brought them up right next to each other. And what can happen is that the carboxyl group on the aspartic acid to the left can form a chemical bond with the amino acid on the right. One of the oxygens and a couple of the hydrogens fall off and they stick to each other, and you're left with a molecule of H2O, which you might know is water, good old dihydrogen monoxide. And that just goes off and just joins all of the other water molecules in the cell. And what we have shown in green is a chemical bond between that carboxyl on the left and the amino on the right, and that's called a peptide bond. So peptide bonds are specifically the bonds that stick amino acids together, carboxyl to amino. And you'll notice if we wanted to, we could bring in a third amino acid on the right and stick its amino to the phenylalanine's carboxyl. And then we could bring in a fourth and a fifth, and we could keep doing this as long as we wanted to and make, you know, as large a chain as we felt like. All we'd have to do is stick each amino acid's amino group gets stuck to the carboxyl of the last amino acid in the chain, right? Just like we can string trains together to make trains as long as you want, we can stick amino acids together to make chains of them as long as we want. By the way, I picked these amino acids because aspartic acid plus phenylalanine plus a couple of extra atoms stuck on one end is a substance called aspartame, otherwise known as NutraSweet. So you've heard of that because that's what's in diet sodas and things like that. Drinking way too much of it may well be bad for you. I'm not really up on the latest nutritional research, but aspartame itself is basically 2 amino acids stuck together by a single peptide bond. So that's a substance that you actually know about, I assume, that is an example of, well, a peptide bond joining 2 amino acids. Ta-da! Okay, right. What's so important about proteins is the shape that they take. And I've compared this before to the shapes of tools. You know, a knife in your kitchen can do certain things very well because of the shape that it's got. And if you cook, you might know that there are differently shaped knives for different functions. You know, you wouldn't want to use a vegetable paring knife to, you know, carve your turkey or something like that, not unless you were in a real bind. You know, there are differently sized and shaped spoons for different kitchen tasks, different sizes and shapes of pots and pans. And they're all able to work because they've got a particular shape. And it's the same with proteins. Your proteins function because they can take on the shapes that are necessary for them to function the way that they do. We're going to keep looking at this after the second exam, by the way. So do not do a memory dump after the second exam. We're almost at the top of the mountain right now. I always think of the middle part of the class where we do the genetics as being like climbing a mountain. We're almost at the top, and once we're on the top, we'll be able to look around and see all kinds of really cool stuff. So here we go up this slope. When we talk about the order in which you put amino acids together, that is a protein's primary structure. But you never make, whenever you stick a bunch of amino acids together, one of the things that happen is that the chain begins to want to stick to itself. If you've ever had to handle very long pieces of sticky tape when you're wrapping a Christmas present or something like that, this is a little bit like what happens. Just like a long piece of tape will tend to stick to itself if it gets the chance, chains of amino acids will stick to themselves given the chance. And a chain of amino acids will typically start folding caused by attraction. You could think of it as similar to static electricity attraction caused by attraction between electrically charged atoms. And we call that level of folding secondary structure. Secondary structure affects only a part of the chain, but the entire thing can then fold up caused by electric attraction or repulsion between R groups, and we call that tertiary structure. And then very often you can have several amino acid chains that have to fit together to make one functional molecule. There's a protein in your blood cells called hemoglobin that is responsible for transporting oxygen from your lungs to the rest of your body. For your hemoglobin to function, it's not made of just one chain of amino acids, it's made of 4. Each of those 4 has to fold up into the right shape, and then all 4 have to stick together. And if they don't stick together in the right shape, you end up with faulty hemoglobin. You could end up with, for example, sickle cell anemia. And we call that quaternary structure. Okay, so if you could see a chain of amino acids, it might look like this. We've taken the carbon atoms and represented them as little green sticks with oxygens in red, nitrogens in blue, and hydrogens in white. This is just one way of building a model of atoms. So that's a stick model of what a simple chain of amino acids would look like. And a single polymer of amino acids is often called a polypeptide, or sometimes just a peptide for short. So here's the primary structure of a protein called insulin. If you don't make this or your body can't respond to it, you have diabetes. And to make insulin, cells in your pancreas have to take a methionine and stick on an alanine and then add a leucine and a tryptophan and another methionine and an arginine and a leucine, proline, leucine, alanine, tryptophan, glycine, proline, aspartic acid. I take it you're writing all this down. I'm actually kidding. You certainly do not have to write this down. This is just the primary structure of insulin. Every one of these funny looking names is one of the 20 amino acids, and they have to be assembled in that order from beginning to end. Not the most fascinating thing you'll ever see, but if your body does not have the ability to put 20 amino acids together in exactly this order, you're in trouble because you will no longer be able to make functioning insulin. Are we almost done? There. So that is primary structure. Secondary structure, certain atoms in amino acids that are close to each other in that chain tend to attract each other, and they make the chain bend and fold. So when particular atoms that are near each other in the chain attract each other, then portions of that chain, local areas of that chain, begin to fold up or twist up. And there's one type of secondary structure. It's called an alpha helix. Not the same as the double helix of DNA. We're not showing you all of the atoms, but all of the amino acids fall along that thing that looks like a twisted red wire or a twisted phone cord, except none of you know what those were. So that's one example of secondary structure. A portion of a chain of amino acids will twist up into an alpha helix. The other possibility is that a portion of a chain will, instead of twisting up, will kind of zigzag back and forth and create a structure called a pleated sheet. It's not really obvious from this picture, but if you could see all the atoms spread out, they would take on a shape somewhat reminiscent of like you taking a sheet of paper and folding a fan out of it and then unfolding it so that it's got these folds in a kind of zigzag pattern. So secondary structure affects a portion of a chain of amino acids, and portions of a chain might kink up into alpha helices, or they might zigzag up into beta sheets. Or in some cases, they don't form any obvious regular structure like this, but alpha helices and beta sheets are examples of secondary structure. Now, tertiary structure happens when R groups attracting or repelling each other make the entire protein fold up. So this happens to be a protein that's found in muscles. It's important for transporting oxygen within your muscles. It's called myoglobin. It's responsible for the dark red color of muscle, or for that matter of, meat. If you buy raw meat, it's dark red, beef anyway. A lot of that is because there's lots of myoglobin in beef. And myoglobin is one chain of amino acids. Parts of that chain fold into alpha helices. That's the secondary structure. And I'm counting 7 alpha helices here. This is the way this is diagrammed. You're not seeing individual atoms. The alpha helices are the parts of the chain that look like phone cords. But the tertiary structure is the way that the entire chain folds up, right? So secondary structure affects part of a chain. Tertiary structure affects the whole thing. This is a protein made by cells in the intestine that's made mostly of beta sheets. The way it's diagrammed here, it looks like an extremely colorful ribbon. That ribbon represents beta sheets of amino acids. And then this is one that actually has both. Don't worry about what this thing does. It's called triose isomerase. The very fine red and green lines that you see are indications of where the actual atoms are. But if we step back a little bit and don't focus on those, you can see parts of this protein are diagrammed in red. Those are the alpha helices. Parts of this protein that are diagrammed in green, those are beta sheets. And then parts of the protein that are neither are shown here in purple. So the entire thing that you're seeing there represents the tertiary structure of the protein. Here's some more examples of tertiary structure. In this case, we put the alpha helices in blue. The beta sheets are in red. There's no real convention for which is which, so the colors do shift from slide to slide. That's the way it goes. The regions that aren't either helix or sheet are in gray. And then finally, quaternary structure, you have several polypeptides. Each one of those has its own tertiary structure, and when they all join together, what we have is quaternary structure. So over there at the top left, that's a single chain of amino acids. You can see it's got a lot of helices in it. That's called globin or hemoglobin. But for hemoglobin to work, you have to take 4 of those and stick them together into a nice, neat, compact unit. And that is hemoglobin's quaternary structure. So for your hemoglobin to work, you have to make 4 chains of amino acids. Each one of those has to have the right primary, secondary, and tertiary structure. And then the quaternary structure describes the way that all 4 fit together. Here's another example. This is an extremely important protein in a long process that we're just not going to have time to get into, but it's critical for extracting energy from the food that you eat. You're basically able to run your metabolism because you have enormous amounts of membrane-bound ATPase enzyme, which is what this is, in certain structures inside your cells. And for it to work, it consists of 7 separate polypeptides, and in this case, each one is color-coded. You have 6 that are kind of shaped a little bit like cake slices that make this kind of rounded structure, or I may be dating myself, but it reminds me of Trivial Pursuit game pieces. If any of you have ever heard of that. And then in the seventh polypeptide is coded in red, and you can see that makes kind of a central stalk, a little bit like the stem of a mushroom. So each one of those different colored squiggles represents a chain of amino acids that has had to be assembled in a particular order. Each one of those chains, parts of it have coiled up or folded up into alpha helices and beta sheets. That's the secondary structure. Each of those chains has then folded in on itself to take on an overall shape. That's the tertiary structure. And all 7 have to fit together to make this thing that looks a little bit like a very psychedelic mushroom. That's the quaternary structure. And if any of it goes wrong, there's a good chance that this protein won't function the way it's supposed to. In this case, if this protein doesn't function the way it's supposed to, the cell probably would not even survive. Losing functioning ATPase is just not the kind of thing a cell can recover from. Okay, this has all been a little bit abstract, but let me try to bring it down to earth in the time that we've got. This is a diagram of a protein called gliadin. It's actually one of a family of proteins called gliadins, and it's found in plant cells, in particular in grains, notably wheat, but also in some others like barley, for example. So that's a single molecule of gliadin. The alpha helices have been diagrammed here in red. The beta sheets are in blue. And the parts of the chain that are neither helix nor sheet are shown in purple. Gliadins join together with themselves and with other proteins called gluteinins. And collectively, they make up what we call gluten. Gluten is a mixture of proteins that's found in plant seeds, in particular in wheat and barley and rye and things like that. And I'm assuming you've heard of gluten because there are people that react very badly to gluten. You can get a lot of gluten-free products at the supermarket these days. We have to in my house because my wife is very sensitive to gluten. She, let's just say, feels really bad if she accidentally ingests some gluten. You don't have to tell me, but some of you might be gluten sensitive. There's various, different conditions, different types of being sensitive to gluten. So anyway, that's what gluten is. It is a mixture of proteins inside wheat seeds and some other grains. Here's what gluten does. You can, if you start with a variety of wheat that has been artificially selected, remember artificial selection and all that? Some varieties of wheat have been artificially selected to have high protein, to make lots of gliadins and gluteinins. If you take that wheat, grind it into flour, mix it with water, and maybe add yeast or salt or whatever it is you're doing, mix it into a dough, and then you take that dough and you knead it, squeeze it and slam it together and squeeze it and slam it together. The mixing with water and the kneading cause all of those gliadins and gluteinins to assemble into an enormous quaternary structure that can fill all of the dough. You still can't really see that quaternary structure, but the effect of this is you have this network of stuck together polypeptides all through the dough, and that tends to make the dough extremely stretchy. That's how this guy can pick up a lump of pizza dough and spin it. And any of you ever seen people doing this? You can take pizza dough and spin it around in the air to make a perfectly round and flat crust. And that's possible because pizza is made with high protein wheat in a dough that is kneaded and encouraged to have all of those proteins assemble into that giant quaternary structure that makes the whole thing hold together. Your pizza crust holds together because it's got many millions of protein molecules that are all interlocked into a network that fills all of the dough. And so the whole thing is going to be very stretchy. And the finished product depends on how thick it is and how fast you bake it. But if you think of a nice chewy pizza crust, that stretchiness is preserved even after you've baked it. If you have a chewy, pizza crust with a lot of spring in it, I guess that would be Chicago style pizza crust. New York style tends to be thinner, but you're college students, you've eaten pizza, you know what I'm talking about. The same for flour that is best for breads, especially styles like French bread or something like that have to be maybe crusty on the outside, but chewy in the inside. Those dinner rolls that I used to eat in my grade school cafeteria that were so chewy, we could mold them like clay and, make little bread animals and things like that. I don't know if you ever did that, but that would be very high protein flour. And it has that chewy property because of the formation of ginormous quaternary structures. The flip side is that if you take a variety of wheat that has been selectively bred by artificial selection to not have as much protein, that is not going to form that kind of stretchy network in the dough. And in fact, we don't even really talk about dough. This is the type of flour that's used for cakes and pastries, right? And these tend to be more delicate, more crumbly, right? I assume you've never had a birthday cake that you've bitten into and then could, really stretch, right? You can't really gnaw on birthday cake the way you can pizza crust. And that's the reason. Cakes and pastries are made with a lower protein variety of wheat. And you also don't need them. You mix the batter. For some things like biscuits, you might need them a little bit, but you don't want to overdo it. And so that quaternary structure never develops and you have a more crumbly baked good. So now you know. If you buy all-purpose flour, that's kind of in between, and it'll do okay for just about anything. But if you want to make really delicate, crumbly pastries or, really thick and chewy, stick to your ribs pizza crust, you're going to want to get the specialized flour because flour for crumbly things has less gluten in it and can't really build up those kinds of quaternary structure networks that high protein bread flour can. Unfortunately, at my house, we have to get gluten-free flour. Cake flour has less, but we can't have any. And I don't know how many of you have eaten gluten-free baked goods. They have to add other things to the flour to simulate the way that gliadins and glutenins make that quaternary structure. And there's nothing that, there's no real perfect replacement for that, which is why most gluten-free bread is still going to be more crumbly and less stretchy than regular bread. So yeah, if you actually buy some gluten-free bread sometime, I actually bake some that's pretty good, but we can't quite get the same stretchy texture that you can with a nice gluten-rich bread flour. So it goes. Anyway, so that was my attempt to try to take all of this stuff and tie it into something you might see. Every living thing makes a ridiculously large number of proteins. You make something on the order of 25,000. I forget the exact number. Different cells in your body make different proteins. Proteins do a ridiculously large number of things. Proteins work because they take on a shape, and the shape of a protein depends on the order of the amino acids. I'll add one last thing. You can alter the shape of a protein molecule by heating it, subjecting it to acid, or maybe doing other things. We call this denaturing the protein. If you denature a protein, you don't break it apart, but what you do is cause it to change shape. Very simple example of denaturing a protein would be frying an egg. The so-called white of an egg is mostly made of a protein called albumin. And when it is functional in the egg, it is, well, it looks like raw egg whites. It's transparent and very runny. If you take that egg white and subject it to heat, as you do if you fry an egg, you can see it going from being, transparent and runny to white and solid. Now, you're not breaking the albumin down unless you're burning the egg. But what that heat is doing is not breaking up the protein, but it's making it change its shape. And once it changes its shape, it no longer has the physical properties that it did. It goes from being runny and transparent to solid and white. Another example of denaturing proteins. Your hair is made of protein. There are chemical treatments that will change the shape of hair molecules of the protein keratin that are in your hair. You can change the shape with heat and chemical treatments. And if you change the shape of the molecules of keratin that make up your hair, you change the shape of the hair shaft. And that means you change the texture of your hair. And I've just described getting a permanent. The reason perms require heat and smell really bad is that what you're doing is changing the shape of the protein molecules that make up every hair shaft. And once you do that, you can change the texture of every hair on your head. That's the way perms work. If you want another one, you could go home and do this. Heat some milk in a saucepan over the stove. Don't let it get really boiling, but when it's about to boil and just starting to steam, throw in a bunch of lemon juice or a bunch of vinegar, stir it really well, take it off the heat and let it sit. And what you'll find is that it curdles. And in fact, you could strain out the liquid, save these solid lumps that appear in the milk, press them together, and you'd have a type of cheese. Queso fresco in Mexican food, or paneer in Indian food. Or curd cheese in English is the old word for it, or cottage cheese. All of those are made of milk proteins that are usually dissolved in the milk and just floating around. But when you change their shape by hitting them with acid and heat, they clump together and they no longer stay dissolved in the milk, and they fall out of the milk and make these big lumps. I actually used to demonstrate that in this lecture. It's not really possible for me to do it today, but it's pretty easy to do. And that's an example of denaturing a protein. You're not breaking it up, but you're changing the shape of the protein. And the shape of the protein depends on the order of the amino acids, and the shape of the protein determines what the protein can do. And last but not least, your DNA carries the instructions for assembling a protein of in a particular order of amino acids. If you inherit faulty instructions, you'll make a faulty protein, and that can cause all sorts of genetically inherited medical conditions and diseases. And we will talk about those later, but I've got 10:50. So by the power vested in me by the state of Arkansas, I'm going to stop the recording now.